Okay, so we are working in the Relationships and Biodiversity notes packet. And there are lines next to each of the slides so that you can take notes on anything that I say that's important. Okay, so first thing, you should have already read the first page of the lab. And if you didn't, just keep this in mind when we're going through it in class, that cure-all, the word that they're using, is fiction, not true, okay? So cure-all is a fictitious plant extract, which means it's like an oil or something that they get out of a plant that they can use to make medicines and things like that. So it's fake, and they only made it up for the purposes of this lab. Got me? Cool. All right, so what does this lab entail? You're going to have to do 7 different tests. And the room will be set up so that there are stations for each of the different tests. I'm recording. Thanks. That was Jaden Gordon. Cameo appearance by Nick Silva. Okay. So there are going to be 7 different tests that you have to do. And there will be different tables for different tests. We're going to try to break it up over 2 or 3 class periods. And it's going to be up to you to keep track of where you're at for each of those things, okay? Let's see. So it says 7 tests that look at the physical, chemical, and microscopic characteristics. So you're going to be looking at the structures on different plants, and you're going to be looking at the chemical makeup of the plants, meaning like what kind of genes do they have. And then you're also going to be looking at microscopic pictures of the plants. Okay. We're going to compare the data, and we're going to try to see if we can identify an unknown species. Okay, so the first test that you're going to do is structural characteristics. So if you look at the different leaves that are on the screen, Botanicurus, species Z, species Y, and species X, they all have specific features about them, okay? So the first one, the first question says, which leaves most closely resemble the leaves produced by Botanicurus? So you're going to look at the leaf for this one, and you're going to look at the leaf for species X, Y, and Z, and tell which one you think is most closely related based on what they look like.
My test is in Spanish.
Hold on. All right, so once you've decided which one that you think looks most similar or has similar characteristics to Botanicurus, then you're going to record those observations in the data table. So your data table for those observations is on page where it says test 1. Okay, so it's on page 8 of the lab. Table 1, comparison of Botanicurus and species X, Y, and Z. Okay? Next thing that you're going to take a look at are the seeds produced by each one of those plants. So here's the seeds that are produced by Botanicurus, and then we have species X, Y, and Z. You're going to take a look at the seeds and see what structural characteristics they have that are similar to each other. And the same thing, on page 8 of your lab, you're going to write down the structural characteristics of each one of the seeds. Okay, test 3. You're going to be provided with 4 different pictures. We're not actually going to make the slides because that would take too long. So you're going to be provided with 4 different pictures of Botanicurus, which is the known species, and then species X, Y, and Z. And again, you're going to compare the structures that you see in the microscopic pictures to the structures that are in each one of the species. And again, on page 8, under structural evidence and microscopic stem structure, you're going to write down your observations. Okay? All right, test 4 is a little bit more involved. You need to do paper chromatography. So you probably have done chromatography maybe in 6th grade or 7th grade. And basically what you're doing is you're putting a spot of pigment from each one of the plants onto a piece of filter paper or chromatography paper, and when you dip it into a solvent, that solvent gets carried up the paper and it drags out all of the different pigments that are in the plant. And then you're going to compare. Well, first you're going to record your observations for what colors you see and how far up they traveled, and then you're going to compare the known species, Botanicurus, to the unknowns. Okay? Test 5. We're going to use an indicator. We've done indicators before, okay? Indicators like what we did with starch and iodine, and what we did with Benedict's solution and Burette's. Indicators will change color or do something else to indicate to you that something is present. So you're going to use the indicator on the known species and on each one of the unknowns, X, Y, and Z, and you're going to record your answers. So you're going to be seeing, in this case, the indicator will fizz if something is present. Okay? On page 8 is where you're going to record the results of that. Test 6. We're going to use a simulated gel electrophoresis. So just like what we saw online the other day, we're going to do an example of an electrophoresis for Botanicurus, and it's showing you that this one has a strand of DNA that's this long, then another strand, then another, then another. And you're going to compare the strands to see how similar they are. The lines indicate where the strands of DNA are cut. Okay, on page 9 of the lab is where you're going to be recording information like this. You're going to be writing out the actual base sequence for each one of the strands, and you need to put it, in the right row of the chart to show how long it is, how many bases it is. Okay, so if you take a look at the top corner, it says number of bases, and then Botanicurus has these bases in it. This is just like a DNA fingerprint, and you're going to use it to compare. All right, last but not least, we're going to take a look at the molecular evidence. Molecular means molecules, and we're looking at nucleotides. Those are the molecules that we're looking at. So we're checking to see what Botanicurus has and what amino acids it codes for, and then we're going to compare that to the other species, X, Y, and Z. Okay, you're going to use your codon chart, and that's going to help you to figure out what amino acids are coded for by each one of the plants. You have a copy of that on the back of your lab. Okay, so the main goal of this is to figure out what's the closest and most probable alternative source for cure-all. So there's a story that goes with this, and you're checking to see which plant, is it species X, Y, or Z, that has most of, if not all, of the same structural, microscopic, and molecular characteristics that Botanicurus has. Ta-da!